Okay, so I keep using this word, resolution or resolving power. The definition of resolution or resolving power is the distance between 2 points that can still be clearly seen as two separate points. If you can imagine looking at the Earth from above, from the space station, I think you can imagine that if there was a bright light shining in Richardson and a bright light shining in Garland, that you, from the space station, you wouldn't be able to tell that those were two separate lights, because from your viewpoint, they would just merge into one, assuming you could even see them at all. From that amount of distance, you wouldn't have resolution. I don't know what the distance is between the center of Richardson and the center of Garland, let's say 5 miles. You wouldn't have a resolution of 5 miles, but you would probably be able to see if we had a bright light shining in Dallas and a bright light shining in Fort Worth from the space station. You might be able to see those as two separate points. Even that would be pretty close. But let's say from the space station, you could say that the light from Dallas and the light from Fort Worth is just barely, but two separate points. That would mean that the limit of your resolution is about 30 miles, whatever the distance is between those things. So the limit of your resolution is called your resolving power. And so I give you this example. If the resolving power of microscope A is 100 nanometers, that means the smallest distance that exists between two objects that can still be clearly seen as two objects is 100 nanometers. But the resolving power of microscope B is 20 nanometers, so closer together, you can still see them clearly. Which microscope has better or more resolution? Actually, it's the smaller number, because if you can clearly see using microscope B two things that are only 20 nanometers apart, but you can still see them clearly apart, separated, in microscope A, you can only see them separated if they're 100 nanometers apart, then B is stronger. B has better or more resolution. So that is kind of counterintuitive, because it's the smaller measurement. The resolving power that's better or more is actually the smaller measurement. If you could see Garland and Richardson as separate using a telescope A from the space station, and you could see Garland, you could only see Garland and Fort Worth as separate using telescope B, then that would mean telescope A had more resolution, more clarity, could see more detail at a smaller level. That's what resolution and resolving power is. So when we're using a microscope, we need to be able to see things clearly. We need to be able to distinguish things as separate. And contrast gives us a lot of assistance in that. When there is contrast, meaning different intensity between two objects or the background, you can see items more clearly and more clearly as separate. And so we use stains to increase contrast. When we look in this example on A, on the left side, you see some cells that are not stained. They turn a little gray or a little kind of a green, if just because the way the light passes through them, but they're definitely hard to see. By putting a stain on them, a simple stain in this case, just one stain of purple, you can see on B how much easier it is to see objects. All right. And so we say that stain increases contrast. You can also use your phase setting on your microscope, and that gives you better contrast typically. So a combination of stain and phase can be used, but phase can be used without stain. So sometimes that's desirable. So on this slide, we just review how to make a specimen for staining. We make a smear. You put a liquid culture in a thin layer on a slide. You let it air dry, and then you heat fix it by passing it through a flame, a Bunsen burner. Then you put the stain on. Most bacteria have acids in their cell walls. We talked about teichoic acid. There's other kinds of acids in cell walls. Mycolic acid is another one. And so the basic dyes are the ones that we use when we want to stain bacteria, because basic dyes attach to acidic things. And since bacteria have acids in their cell wall, we use basic dyes to stain the cell wall. So I mentioned acidic dyes first, but we really don't use those in staining bacteria. So we choose the one that's appropriate for the chemistry of the organism that we want to stain. So some simple stains, single stains, can be used. And these are really easy to use. Once you heat fix your spread, your smear, you're going to then put a dye like crystal violet or safranin or methylene blue. Crystal violet is a purple color, safranin is kind of a peachy pink, and methylene blue is a blue, a nice deep, pretty deep blue. And then you let it set for 1 minute and then you rinse with water. And that, when you look at that under the scope, you can see very good contrast. So you can estimate the size of your cell, you can identify the shape, we'll talk about that in a minute, and arrangement. A differential stain, that term refers to a procedure that uses two stains or two dyes, and you're going to be able to distinguish, if you have a mixture of cell types, for example, you could distinguish them using a differential stain. Common differential stains are the gram stain. So in a gram stain, you use two stains. You use crystal violet, that will stain the gram positive cells, and you use safranin, that will stain the gram negative cells. But you also have to do a certain destaining. There's a couple other chemicals involved to make that happen. Then we have something called the Acidfast stain. The Acidfast stain is specific to a type of bacteria called Mycobacterium, because Mycobacterium, the most famous one is Mycobacterium tuberculosis. That one is gram positive, but it doesn't hold the crystal violet very well. So typically, if you suspect a tuberculosis infection, you would test it with an Acidfast stain. It uses slightly different types. It uses different types of stains that attach better specifically to Mycobacterium. And similarly, the endospore stain, that involves a chemical called malachite green, which stains endospores. You'll remember those are those really tough structures that form in certain types of bacteria, specifically the genus Bacillus with a capital B, Bacillus, and the genus Clostridium. And so an endospore stain specifically would identify if it's a positive result. If you see endospores at the result of that, at the end of that stain procedure, you could then narrow down your identification of the specimen to one of those genera. And this picture just shows, you don't have to know the entire process for lecture, but for lab you will, so probably makes sense to go through it. You heat fix, you smear your specimen on a slide, air dry and heat fix. Then you put the crystal violet on, rinse with water, do some iodine, rinse with water. Then you rinse with an ethanol acetone solution pretty quickly. And what that does is, at that point, only the gram-positive cells are still able to hold on to that crystal violet. Then you counter stain with safranin so that anything that didn't get stained with the crystal violet picks up a pink stain. And so at least you can see it, but you know it's not gram positive. So at the end of the gram stain, you would have purple cells would be the gram positive ones and pink ones would be gram negative. And this just shows an example of the Acidfast stain. Now I guess you can hypothesize who Nielsen, sorry. Those are the last names of the people who invented this. but you don't need to remember their names. We're just going to call it the Acidfast stain. In this stain, the pink ones are the mycobacteria. So only the pink cells are the mycobacteria, and then anything else that's there gets counterstained blue. In this slide, it looks a little bit purple, but when we look at the real one, it's hot pink for the mycobacterium, and then anything else is just blue. And then the endospore stain, what we call the vegetative cells, are pink from the safranin, but the endospores are green. So that malachite green really tells us if there's spores. In this picture, there are. Some special types of stains that we also do, again, keeping in mind that these are all trying to identify characteristics of our specimen. You can do a flagellar stain, and you can see flagella. So this would be a really good result of Proteus vulgaris. So Proteus has a peritrichous arrangement of flagella, and you can see that with using this stain. So that's called a flagellar stain. It's like a simple stain. You just put a stain on the cells that particularly stains the flagella very well, because not every stain sticks to that structure as well as other stains. The negative stain, which is shown here on the right, is where you basically what you're doing is you're putting a stain on the slide that stains the background, just the glass part, just the nothing. And then if the cell has some kind of glycocalyx, so you'll remember that would mean capsule or slime layer, then that will show up as a clear space around the cell itself. So around the part of the cell where the cell wall is. So when you see these clear spaces, that means there is some kind of glycocalyx. So that's called a negative stain because you're staining the background more so, and the capsule or the slime layer does not stain. It shows up sort of clear. All right. Changing, well, we're ultimately, all of this is about identification of an organism, and you have to know a little bit about how classifications are set up in biology. And you may remember that Linnaeus, he started with classifying things in pretty broad groups. Plants, I think he did plants and animals were his main two groups. And he used what we call the binomial nomenclature, which was genus and species. But really, we built on that, and we now have more than just genus and species. We have domain, kingdom, phylum, class, order, family, genus, and species. You do need to know these levels in order. So sometimes students use a pneumatic device. That doesn't sound right. Mnemonic device, or a sentence such as, did King Philip, with a P-H, Philip, come over for good spaghetti? And that would help you to remember them in the right order. So